Hi everyone, this is lecture 2, Chemistry of Life. And so this is where we divert a little bit between the chapters of the textbook. So our official adopted textbook is Campbell Biology. And so this is going to span chapter 2, 3, and 4 from the Campbell Biology textbook. In the OpenStax textbook, if you're using that one, because it's cheaper, again, easy, this is all kind of condensed in OpenStax in chapter 2. So it's still the same concepts, it's just kind of how they organize information is a little bit different. So we're going to cover the same big picture concepts. And so Campbell Biology, if you're using that textbook, it goes into more detail about these topics, and that's why they each have their own chapter. But I think you are okay just reading the summaries for each of these chapters. So you don't have to read all of chapter 2, for example. I think, just reading the summary at the end of the chapter 2 chapter is good. And then the summary for chapter 3 and the summary for chapter 4. And then, again, it's the same info in chapter 2 of OpenStax. It's kind of all condensed. And so this is going to be an overview of chemistry, which is really important to biology. But also you're going to get a lot of extra detail that is, you're going to cover a lot more about in chemistry. So if you are a biology major, you're going to have to take chemistry at some point. Yeah, it is important. So for this lecture, it's going to be broken up into 3 kind of sections. So chemistry basics, and this is the chapter 2, water, chapter 3, and then carbon, chapter 4. And again, it's still all the same stuff that's condensed in chapter 2 of the OpenStax book. So chemistry. Why do I have to learn chemistry to learn biology? Because it's all related. Everything is made of atoms, including us, including all living things. And so even though chemistry is its own field, there's a lot of overlap between chemistry and biology. And especially in this class, when we're going to talk about like molecules and cells, we're going to talk a lot about different kinds of atoms and elements. And so it's all related. Molecules are made of atoms, and that's what chemistry focuses on. And so it does, blend. So organisms are composed of matter. Matter is anything that takes up space and has mass. And matter is made up of elements. So anything that has mass and takes up space is made of elements. In element, just to define that, you may have heard of, the periodic table of elements, but if you haven't taken a chemistry class or it's been a long time, this might be, a refresher. So an element is a substance that can't be broken down to other substances by chemical reactions. A compound is a substance consisting of 2 or more elements in a fixed ratio. And a compound has characteristics that are different from those of like the elements that make it up. So this is an example of a combination of 2 different elements, so sodium and chloride. Whoops. So each of those is a different element, but when they're combined in a compound, the compound is sodium chloride, which is table salt, and it's very different, as you can see, from sodium and chlorine. So a compound that is 2 or more elements in a fixed ratio has characteristics that are different from those of each of its elements. And so the elements of life, there are some elements that are most common in living things. So about 20 to 25% of the 92 natural elements are required for life. And so these are considered essential elements because they're essential for life. And of all of those different elements that are required for life, the ones that make up the most of it are carbon, hydrogen, oxygen, and nitrogen. Those elements make up 96% of living matter. And then most of the remaining 4% that makes up living matter is calcium, phosphorus, potassium, and sulfur. And then there are some trace elements that are required by organisms, but Those are only required in minute quantities, and so they're called trace elements, not essential elements. So here's a breakdown of the elements in the human body. Again, the most is oxygen, carbon, hydrogen, and nitrogen. That makes up 96% of your body, including water. And then the remainder is other stuff that's important. So calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium, and then trace elements. So if your whole body is made up of elements, it makes sense we have to study chemistry a little bit, right? So each element, what makes them different? They consist of unique atoms. So atoms are the smallest unit of matter that still retain the properties of an element. So here is the symbol that we kind of show for like an atom. And it's pretty simplified because, as we'll see, there's more to it than just one little circle in the middle. So atoms are made of subatomic particles. So altogether, this is one atom. And then the little tiny particles within an atom are subatomic particles. And so there's these other smaller little pieces. So there are three kinds of relevant subatomic particles. So neutrons have no electrical charge. They're neutral. Protons have a positive charge. And then there's electrons, and electrons have a negative charge. And so structurally, this is what it looks like. The protons and neutrons hang out in the middle. And so we call that the nucleus of the atom, kind of analogous to like in a cell, there's the nucleus and it has like the most important things in the middle. So the nucleus of the atom has protons and neutrons. And then around the nucleus are electrons. And so they kind of form a cloud. We'll tend to show them like in a circle, but they actually hang out more in like a dispersed area. So a cloud. And so again, you know, protons positive, electrons negative, and then neutrons are neutral. So all these charges kind of add up, like a positive and a negative kind of cancel each other out. And then neutrons don't have a charge. So overall, an atom is neutral. And the things that make each atom an element, that make each element unique are the atoms. So each element is going to have a specific number of protons. So if this atom has 2 protons, it's this kind of element. If there are 3 protons, then it's this kind of element. If there's 4, then it's this other element. Because these subatomic particles can vary. But protons are what make an atom a certain kind of element. Things like the electrons can change. So there can be like an addition of an electron or a removal of an electron, but that doesn't change what element it is. It might change the charge overall, but it won't change what element it is. And then even neutrons there can be one more or less, or, you know, more. And it's still the same element because the protons are the same number, but the neutrons, if you change the number of neutrons, that makes it an isotope. So we'll, you know, talk more about this in the next few slides, but hopefully that overview kind of helps. And then this is the same thing I'm kind of pointing at. So the middle is the nucleus of an atom. Protons have a positive charge. Electrons have a negative charge. There's 2 protons, but there's also 2 electrons, so they cancel each other out, and they're neutral. And so this is actually an element of helium. So because there's 2 protons, this is an atom, a helium atom. If there was only 1 proton and one of everything else, then that would be a hydrogen. So like I mentioned, neutrons and protons form the atomic nucleus. The electrons form kind of a cloud of negative charge around the nucleus. Even though we show it in like stationary and just a circle, they kind of float around as like a cloud. I think there's a picture right here. And so each of these little subatomic particles has mass. You can weigh them in a way. They're very, very, very tiny, but they do each have mass. So neutrons have their own mass, protons have their own mass, and then electrons technically do have mass, but they're so tiny that we kind of don't even count their mass as like the total. So when you add up, well, first of all, the neutron mass and proton mass are about the same. They weigh about the same. They're almost identical, and they're measured in a form of units called Daltons. And then electrons are so tiny that they're kind of just ignored. We don't really even think about their mass. They do technically have mass, but it's super tiny. So yeah, protons have some mass, neutrons have some mass, and then electrons barely anything. And so again, yeah, this is 2 protons, so that makes this a helium atom. And then these are, this is a more realistic depiction of the cloud of electrons. So atoms of the different elements will have different numbers of subatomic particles. An element's atomic number, what makes it that element, is the number of protons. So each element is going to have a different atomic number because it has a different number of protons. Each element also has a different mass because it has a different number of subatomic particles. So an element's mass number is going to be the mass of the protons plus the mass of the neutrons. And you just add that up together, and that's the mass number, the atomic mass. Because, again, electrons are so tiny, we kind of don't even need to factor them in. So atomic mass can be approximated about the same as the mass number, protons plus neutrons. Now, say again, there's some variation in the number of neutrons, that is going to lead to different isotopes. So all atoms of an element have the same number of protons, but they can differ in the number of neutrons. So isotopes are 2 atoms of an element that have a different number of neutrons. And then radioactive isotopes decay spontaneously, giving off particles and energy. So if you've heard about, things like radioactivity, it's kind of, relevant to this. So radioactive isotopes are often used as diagnostic tools in medicine. So again, another way that chemistry plays a part in medicine is like the tools that we use. So we use radioactive isotopes to help through certain diagnostic techniques. So for example, radioactive tracers can be used to track atoms through metabolism. So as something kind of breaks down and gives off energy, we can track how much energy there is, and that kind of tells you how long it lives or how long this metabolic process takes. Or if you want to track the pathway of something, so these tracers can be used to track atoms through metabolism. They can also be used in combination with sophisticated imaging instruments. So for example, PET scanners or PET scanners. They can monitor the growth and metabolism of cancers in the body. So here's a picture of a PET scan. Oops. And so you would use a radioactive isotope to be able to see the cancer's throat tissue in this scan. So that's another application of chemistry in biology. So again, an isotope can kind of like decay and give off energy, and so a parent isotope decays into a different form, into its daughter isotope at a fixed rate. And so that rate, we can know the half-life of it. So like with this amount of time, it's given off half this much. So a parent isotope will decay into the daughter isotope at a fixed rate, and that's expressed as the half-life of that isotope. And so in another application of chemistry in biology, there is radiometric dating. So scientists can measure the ratio of different isotopes over time. Like, okay, this decayed into this and that decayed into this version. So scientists can measure the ratio of different isotopes and calculate how many half-lives have passed since the fossil or rock that's being studied was formed. So that's another application. And then it depends on whatever isotope you are looking at. You know, they're going to have different half-lives. So the half-life values can vary from seconds to days for some isotopes to billions of years for others. Okay, so that was kind of focusing on if there's differences in neutrons. So let's focus back onto the atom and instead on electrons. So electrons play a part in energy, and they have energy. And so energy defined is the capacity to cause change or to do something. And so there's different forms of energy. There's potential energy, there's kinetic energy. So potential energy is the energy that matter possesses because of its location or structure. And matter has a natural tendency to move towards the lowest possible state of potential energy. And so in an atom, the electrons of an atom differ in their amounts of potential energy based on how far they are from the nucleus. There's actually more than one ring that electrons can hang out in. And so, yeah, the electrons of an atom can differ in how much potential energy they have based on their distance from the nucleus. So changes in potential energy of electrons can occur only in steps of fixed amounts. So I will show a figure in the next couple slides. So electrons are found in different electron shells, and they each have a different average distance and energy level. So here's a picture. So here is the atomic nucleus in the middle, and we're just kind of zooming in on a quarter to show you the shells. So there's different shells of where electrons can be. So the first shell is closest to the nucleus of the atom. It has the lowest amount of energy. There's a second shell that's a little bit further from the nucleus. It has a higher energy level. And then the third shell is the highest energy level. So depending on where the electrons are, that kind of dictates how much energy that electron has associated with it. And so you can kind of, assign different levels of energy to the different electrons. Sometimes if you wanted to push an electron further out, energy is absorbed because you have to put in energy to move it that way. And then opposite, if you wanted to move an electron down, some energy is kind of released or lost that way. So this, you'll get a lot more of this in chemistry. Don't worry about this too much yet. The most important part is the valence of an electron. So an atom that has an incomplete outer shell is reactive. So there's these 3 different shells, and they can each hold a maximum number of electrons. So the first one, for example, can only hold 2 electrons, and then you would shove them to the next shell. And then this one only holds 8, right? Right? 2, 4, 6, 8, yes. So this is showing you, okay, the first shell can only hold 2. The next shell can hold 8. 2, 4, 6, 8. And then the next one can actually also hold 8. 2, 4, 6, 8. So, if we go back to this idea of shells, if an atom has an incomplete shell, whichever one is the one that's the most filled in, if it's incomplete, it's either way, it's called its valence shell. So the one that's like furthest out and most complete is the valence shell. And so if it's not complete, it doesn't have the maximum number of electrons there, it kind of wants to interact with other stuff and fill that shell. So whichever one is the like outermost shell of this atom is the valence shell. If the shell is incomplete, then it's pretty reactive. And so, yeah, how the electrons are arranged in the shells determines the chemical behavior of an atom. And this first point is kind of saying the same thing in different words. The chemical behavior of an atom is determined by the distribution of electrons in those electron shells. Like if the shell is full, then this atom actually doesn't really want to hang out with other stuff. But if it's not full and there's spaces that it wants to fill with electrons, this atom is going to be more likely to interact with other stuff. And so the periodic table of elements is a way that we organize information to actually kind of inherently show how much reactivity there is based on these electrons. So yeah, the periodic table of elements shows the electron distribution for each element, even if it doesn't show you in the same way that this one literally shows you. It's implied. That's how it's organized. So the left to right sequence of elements in each row corresponds to the sequential addition of electrons and protons. So this will make more sense in a second. So in the very top row of the periodic table, it's corresponding to how there's only 2 electron spaces in that first shell. That first shell can hold up to 2 electrons. So if there's only 1 proton and 1 neutron and 1 electron, this is a hydrogen. If there are 2, there's 2 protons, 2 neutrons, and 2 electrons, this is a helium atom. And so this is, telling you that the atomic number, again, is the number of protons. The mass is the protons plus the neutrons, and so it's going to be basically double this. And then, so yeah, this is showing you the electrons. And so if this shell is full, if there's another proton and another electron, the electron's going to go to the next shell. So this is lithium. And so then the next shell has a total of 1, 2, 3, 4, 5, 6, 7, 8 electrons. So if there's a total of 10 electrons, 2 are going to be in the first shell, and then 8 in the next shell. And then this one's all pretty full. It doesn't really want to interact with stuff. And then so, if there's more protons and more electrons, it's going to go to the next shell. And so on. So yeah, as you go from left to right, there's 1 electron plus 1 electron plus 1 electron plus 1 plus 1 plus 1. And so this is kind of showing also the behavior of these atoms. So the last column is what are called the noble gases. You might have heard of them. And so they're called the noble gases because they kind of really don't want to interact with other things. Like, they're like, oh, my shells are full. I don't need you. Like, I don't need to interact with anything. Like, I'm good. And so they call them like the noble gases. They're too noble to hang out with anything else. But other stuff that does have an incomplete shell, it's going to be more likely to interact with other things because it's like, hey, let's link up. And then we both kind of fill our shells. And so notice the ones that are pretty common that we mentioned already are carbon, nitrogen, and oxygen. Those guys have incomplete electron valence shells. And so, yeah, it makes sense that they hang out with a bunch of stuff and make up so much of your body. So hopefully that makes sense. So valence electrons are those in the outermost shell of whichever element you're focusing on. And the chemical behavior of an atom is mostly determined by the number of valence electrons. Elements with full valence shells are chemically inert, meaning they're not going to really interact with other stuff. And so we show, yeah, these like rings to demonstrate electrons, but there's different ways to model electrons and how they hang out in an atom. So an orbital is a 3D space where an electron is found like 90% of the time. And then each electron shell consists of a specific number of orbitals. No more than 2 electrons can occupy a single orbital. And then, yeah, atoms interact in a way that completes their valence shells. So it doesn't literally look like, circles. There's kind of more of like a 3D space that these electrons can be at 90% of the time. And those follow kind of specific shapes. So here is kind of what I mean. So that figure that I showed, there's different diagrams to show these things. So electron distribution diagrams are super nice and easy to show, like the number of electrons in each shell. But you can also have this other diagram that shows the electron orbitals. So the first orbital, the first shell, is just kind of like a sphere. The second orbital, so in the second shell, they can hang out like in an orbital, another kind of sphere, or they can also hang out in these other like 3D directions. So they go in different planes. So this is the X, Y, and then Z. So the second shell, they can actually, the electrons can be in these different spaces, in this way or whatever. And then if we were to kind of, look at all of these all together, instead of focusing on each one, this is kind of what it would look like. So the first shell would be kind of in this darker middlemost circle. And then the electrons of the second shell can be somewhere here, or somewhere here, or somewhere here. And so we point this out because this is also going to dictate how different atoms like can form different shapes of molecules. So atoms with incomplete valence shells can share or transfer their valence electrons with certain other atoms. Then that way one atom is completing its valence shell, or maybe it's like sharing and then they're both completing their valence shell but sharing. So yeah, how they behave is going to depend on their valence electrons and other stuff. So these interactions usually result in atoms staying close together, held by attractions called chemical bonds. So there are different kinds of chemical bonds / interactions. There are strong bonds that are like hold them pretty tightly. And then there are kind of like weak bonds or weak interactions. So of the strong bonds, there are covalent bonds and ionic bonds. And then I'm going to break down each one of these 4 bonds on the next few slides, but just to show you an overview. And then the weak interactions include hydrogen bonds and van der Waals interactions. So a covalent bond is when 2 atoms share a pair of valence electrons. So on the right here, there are 2 hydrogen atoms, and they each have 1 proton because, they're hydrogen atoms. And so here we're showing their cloud form. So hydrogen atoms just have 1 electron. And remember, that first shell can hold 2 electrons, so they kind of want to fill up their valence shell. They want another electron. And so they're like, hey, why don't we just share a pair of electrons, the ones that we have, and then that kind of completes the 2 for each of us. So in a covalent bond, the shared electrons count as part of each atom's valence shell. So I think there's another picture too. Yeah, here. So this one's kind of showing you the cloud, but here is another one, a figure with a water molecule. So here's a hydrogen, and again, it only has 1 one electron, one valence electron, and it wants to fill up a second spot. Oxygen actually has two spots that it wants to fill. And so it can bond with this hydrogen. And then now they're sharing this pair. And so to the hydrogen, it's like, cool, I have two. And then to the oxygen, it's like, cool, I have two in that spot. And then by bonding with another hydrogen, it fills up this spot. So now all of its valence electrons are full. This shell is full. And then for this hydrogen too, it's full. So that is how atoms in a covalent bond share a pair of electrons. So in a covalent bond, you can form a molecule. So a molecule consists of two or more atoms held together by covalent bonds. So a single bond is where two atoms share one pair of valence electrons. So kind of like over here. You know, this is a single bond. They're just sharing one pair. But some atoms can also share two pairs. So a double bond shares two pairs of valence electrons. So in this figure here at the bottom, if these two oxygen molecules kind of rearrange stuff and come together, they're sharing two pairs of electrons. And so they technically complete their electron, their valence shell, because now this oxygen on the left has 1, 2, 3, 4, 5, 6, 7, 8. And then to the oxygen on the right, the same thing. Now it has 1, 2, 3, 4, 5, 6, 7, 8. So they're each happy sharing those two pairs. And there's a bunch of different ways that we can talk about or refer to molecules. So a structural formula is used to show atoms and bonding. And so it'll show, you know, the letter that represents each atom or element. So H represents hydrogen. So an H, a line, and then an H shows that there's a single bond. And then an O and an equal sign with another O shows that it's a double bond. But that's more work. You know, it's kind of implied at some level how stuff is combined. Or maybe we don't even care whether it's a single or double bond. And so we can just abbreviate a structural formula with a molecular formula. So for example, if I don't want to be writing H-H, you could just say H2, it's two hydrogens. Or O2, it's two oxygens. Yeah, you're not always going to take the time out to show the single or double bonds. You're more likely, especially throughout this class, you're going to see molecular formulas because we're going to talk about bigger molecules. And so it's just kind of easier to write the molecular formula. So hydrogen is going to always be hanging out with another hydrogen. So it's going to be H2. Two hydrogens interacting, completing their valence shells. Oxygens are always going to hang out in twos, complete each other's valence shells. Water, H2O. In blood, there's two H's, two hydrogens, and then an O for oxygen. And then methane, CH4. There's 1, 2, 3, 4. So boom. Yeah, we're not always going to have the structural formula because usually it only works with tiny molecules. Yeah, so we're always going to usually say molecular formulas. And then here's all these other ways that you can show these structural formulas with the electron distribution diagram, the Lewis dot structure, structural formula. Yeah, so you'll get a lot more of that in chemistry. But let's go back to the kinds of bonds, right? So we were talking about covalent bonds. And so again, if we touch base on covalent bonds, sharing a pair of valence electrons between two atoms. So the bonding capacity of one atom to be able to bond with other stuff is called the atom's valence. Covalent bonds can form between atoms of the same element, like H2 or O2, or atoms of different elements, like H2O. And a compound is a combination of two or more different elements. So that is all assuming that they can evenly share electrons. But atoms can be different. They can attract electrons to different degrees. So sometimes they attract electrons really strongly. Sometimes they don't attract electrons super strongly, and so they kind of lose their electrons. So electronegativity is an atom's attraction for the electrons in a covalent bond. The more electronegative an atom is, the more strongly it pulls electrons towards itself. Even if it's sharing, it's kind of like hogging them. So electronegative atoms are like greedy, like they want to hog electrons. My analogy for this is like toddlers. Sometimes, you can have toddlers that are technically sharing, but they're not actually sharing, Some toddlers are used to sharing, and then, they'll split time equally sharing a toy. But some toddlers are not very fair like that, and so they'll be like sharing a toy. But it's like, okay, I'm going to play with this toy for 5 minutes. You can have it for 1 minute and then give it back. They're like more greedy. They want to hog something. And so that's how electronegativity is also. Like, how much does this want to like hold on to these electrons? So that can result in differences in bonding. So non-polar covalent bonds, the atoms share the electron equally. Electron or electrons, they share them equally. They're good sharers. In a polar covalent bond, one atom is more electronegative and the atoms don't share the electron equally. So unequal sharing of electrons in this molecule will end up causing a partial positive charge in one place and then a partial negative charge in the other place for each atom in that molecule. So here is a figure showing that. So this is water, H2O. And so water is actually super electronegative. It really likes hogging electrons. So because the oxygen likes hogging electrons and they're spending more time with the oxygen, and then remember, electrons are negatively charged. So in this overall molecule, the oxygen is going to have a partial negative charge. That's what this little like weird shape is. It's indicating that there's a partial negative charge. And so again, that's because the electrons are kind of being hogged by the oxygen and electrons are negative. And then on the hydrogens, it's kind of like losing its electrons. They're being kind of shared unequally. And then, so that just leaves the positive charge from the protons. And so now this part of the molecule has a partial positive charge. So this is a polar bond. So think of like the North Pole and the South Pole, or like polar opposites. A polar bond has like this very large difference between like one part's positive, one part's negative. And so here's another figure showing different kinds of covalent bonds. So if there's a polar covalent bond, then one part is going to be more negative and then one part's going to be more positive. And then it also depends on how the atoms can fit together because of their electron orbitals. So they might have different molecular shapes that result in the molecule either being more likely to be polar or less likely to be polar. So water, the way that the atoms want to stay in shape, this molecule is like this, at this angle. Methane, nothing's polar. It kind of shares everything equally. The whole molecule is nonpolar. Carbon dioxide, there is a polar bond between the carbon and each oxygen. But because of the way the atoms want to hang out it's linear, and then that makes the whole thing nonpolar. So there's a lot, there's a lot to chemistry. Yeah. But right now we're just talking about the bonds, just a polar covalent bond and a nonpolar covalent bond. Another kind of chemical bond are ionic bonds. So atoms will sometimes steal electrons. It's so electronegative, it's just going to steal the electrons. So you might have seen that with toddlers also. They're just going to steal a toy, and that's it. So sometimes atoms steal electrons from their bonding partners. They're not really even sharing or pretending to share at this point. They just straight up steal it. So that means those two atoms are not going to have different charges. The two resulting oppositely charged atoms are called ions. And so an ion is any kind of charged atom. So if it's positively charged or negatively charged, we'll just call it an ion. But we can be more specific based on if it's positively charged or negatively charged. So a cation is a positively charged ion, and then an anion is a negatively charged ion. So yeah, if you were to hear just ion, it's some kind of charge, either positive or negative, and then these are more specific. And you can kind of remember it by cation. Cats are cute, and so it's positive, right? One student said positive. And then anion, onions make you cry, right? And so it's negative. So anions are negatively charged. Whatever you have to do to remember stuff, So anions and cations attract each other. And so this attraction is called an ionic bond. They kind of just want to hang out together. So fluorine is very electronegative and just straight up steals an electron from sodium. So then each one is an ion. So a sodium ion, and then a chloride ion. And remember, now that the electron is switched, now this is going to be more positively charged. And then now this is going to be more negatively charged. So this is a cation on the left, and then this is a chloride ion, an anion on the right. And together they kind of just hang out near each other, and so this is an ionic bond that makes sodium chloride hang out together. So compounds formed by ionic bonds are called ionic compounds or salts. So sodium chloride is a salt. It's like the most common salt we know of, but there's other ones too. So they are just compounds formed by ionic bonds, and that makes those other stuff salts too. Salts like sodium chloride, table salt, are often found in nature as crystals. Sodium chloride itself is not a molecule per se. The formula for an ionic compound indicates the ratio of elements in a crystal of the salt. So it's not necessarily a molecule, it's kind of more of a compound. Yeah. Most salts are pretty stable when they're dry, but usually dissociate pretty easily in water, and we'll talk about that in part 2 of this lecture. So here's sodium chloride as a crystal, and it's these two ions just hanging out with each other. They're not covalently bonded, they are ionic bonded by just hanging out together, basically. So those are both pretty strong interactions. Most of the strongest bonds in organisms are covalent bonds. And yeah, so most of those bonds are covalent bonds that form a cell's molecules. But there's also many large biological molecules that are held in their form by weak bonds. So we've been focusing on kind of really small stuff. Next week, we're going to talk about large molecules. And so these molecules are going to be so big that, yeah, they're combined by covalent bonds, but there's other parts of the molecule that get held together by these weak bonds. So it is really helpful to also have weak bonds for a reason. You might think like, oh, why aren't they all strong if stronger is better? Sometimes it's really useful to have weak bonds. So for example, if you think of the DNA double helix, you know, the double helix like that, that's two strands of DNA, this strand, and they're each held together by covalent bonds, but then the two strands are held together by weak bonds. And that's really helpful because there's certain times where the cell needs to unravel the DNA and like split like a zipper the DNA. And so you don't want it to be too strong. It's really helpful to have a weak bond because you can do that and then redo it. So the reversibility of weak bonds can be a real advantage. And there are several types of weak chemical interactions important in organisms, and we'll just kind of talk about two. So hydrogen bonds. A hydrogen bond is an attraction between hydrogen and then something else. So it's usually a hydrogen atom that has a partial positive charge that's on one molecule, and it's attracted to a different molecule, an atom that is very electronegative and carrying a partial negative charge. Oops, this figure I think cut off some text. So here are two different molecules, right? Here's water, which is polar and has a partial charge. And then here's ammonia, which also has its own partial negative and partial positive charges. And so a hydrogen bond with a partial positive charge attracted to the nitrogen on this molecule. This nitrogen is electronegative and it has a partial negative charge, and so this is a hydrogen bond. It's pretty weak, but again, sometimes that's good. So in living cells, the electronegative partners are usually nitrogen or oxygen. So like this water molecule can actually hydrogen bond with another water molecule. So a hydrogen will usually interact with the oxygen of a different one. But this is an example of something else. So another one is van der Waals interactions. These aren't even necessarily bonds, it's more interactions. And you really don't hear much more about them after today. So if electrons aren't evenly distributed, because remember they just happen to hang out in different areas like clouds. So if electrons aren't evenly distributed, they can build up by chance in one part of a molecule. And so van der Waals interactions are attractions between molecules that are close together because of these charges. And altogether, these interactions can be strong. So each of these little tiny hairs on the bottom of this lizard's foot has these kind of van der Waals interactions happening. And so through all of those interactions happening between all of these weird little hair fiber ends and the wall or whatever, that's how they can kind of grip on because of the sum of all of these interactions. And that's kind of it. That's all you'll hear about van der Waals interactions pretty much. And so the way a molecule looks, its size and its shape are really important to its function. Like how is it going to be able to interact with other stuff based on how this molecule looks and it's shaped. So a molecule's shape determines how biological molecules can recognize each other and respond to each other. So one kind of example are opiates, like morphine. So at one part of like an opiate molecule, the shape and size can be very similar to a natural occurring endorphin. So naturally produced endorphins at this one part have very similar shape, right? And so that's kind of how morphine can trick these endorphin receptors to be activated by morphine. So they have similar effects because their shapes are similar and they bind to the same receptors. And so this is another way that chemistry is really important in biology, is like molecules and their effects on their targets and cells. So chemical reactions are the making and breaking of chemical bonds. So sometimes, atoms will hang out together and then form a covalent bond, and now they're a molecule. So that's kind of a reaction. And then if you were to break them and reform them, that's another chemical reaction. So chemical reactions are making and breaking bonds. You'll take some molecules and they'll interact and maybe break those bonds and break these bonds and then rearrange themselves into these. And so what you start with are called the reactants, and then what you make are called the products, which makes sense, right? So all of the atoms on each side of a chemical reaction, nothing gets like destroyed or lost. They're going to be the same number, just rearranged. So I think one textbook kind of compares it to like dance partners. So even if you are in like a dance hall or whatever, and then you change dance partners, the same people are here overall, the same number of people, they just changed partners. So there's still 1, 2, 3, 4 hydrogen atoms. And then they just rearrange themselves, and now there's still 1, 2, 3, 4 hydrogen atoms in the products. And then again, left, there's 2 oxygen atoms, and then on the right, there's still 2 oxygen atoms. They just rearranged. They just broke bonds and then remade bonds and switched dance partners. So the stuff that's on the left of a chemical reaction are reactants. We'll usually show an arrow to indicate that's the reaction happening. And then the products are going to be on the right. So yeah, chemical reactions just rearrange the reactants into the products. And again, it's relevant to biology because in your body and all your cells, a bunch of chemical reactions are happening. So one really big example is photosynthesis. Photosynthesis is a really important chemical reaction where you take carbon dioxide and then molecules of water, and then the sunlight powers the conversion of this carbon dioxide and water, rearranges the atoms into glucose and oxygen. So they're all very different molecules, and this is overall one big chemical reaction that happens actually in a bunch of different steps, but this is the big picture reaction. And then here's another one that's kind of showing the molecules. So if there's carbon dioxide and water, you're rearranging these pieces, and now there's glucose and oxygen. And that's powered by sunlight, because sometimes it takes energy to make a chemical reaction happen. Yeah. And all chemical reactions are reversible. The products of that forward reaction, if you were to flip it, now they're reactants for the reverse reaction. And so if you see an arrow with 2 heads going in the opposite way, that's showing that this reaction is reversible. So a lot of times we'll show it, it's one arrow, but especially if you see 2 arrows, they're kind of more easily reversible. And so chemical equilibrium is reached when the reverse and forward reactions are equal. At equilibrium, like it's kind of going to go one way and then the other, but in total, the relative concentrations of reactants and products don't really change at equilibrium. Okay, so this was a lot of information, and there was some like details, but most of those finer details we could just leave in chemistry. Think of the main ideas. So the main ideas of this section are that matter consists of chemical elements in their pure form and in combinations called compounds. An element's properties depend on the structure of its atoms. And then the formation and function of molecules and ionic compounds depend on the chemical bonding between atoms. So those are the big picture ideas. So try to think about that. And then everything else can kind of fit more into that idea. Hopefully that helps.